our last speaker of the evening is Miguel Sena Esteves, who is actually a pioneer of gene therapy and much of the AAVs. He's going to talk about AAV9 gene therapy for the GNAO1 deficiency. And he comes to us, he's local, he comes from us.
I know. Believe me, it's worse to be local with the traffic, actually. So how do I change presentations? Oh, just keep going. Oh, right, sorry. Okay. I mean, one thing that I realized is that we all use BioRender. Very useful. Anyway, so, okay, I guess the title, right. So AAV, it's, has an interesting history to it. Of being found as a contaminant and becoming, a very useful platform, as well as the unknown impact of doing research that at the time sounds completely idiotic of why would you even do it, and 60 years later actually turns out to be a very important tool, right? So it was discovered, I think it's 1965, as a contaminant of adenovirus. Researchers tend to be a little boring, so they couldn't come up with any better name than telling that it's associated with adenovirus. So that's sort of the history, the humble history of how it started as a little contaminant, and we all know about the SMA and the transformative results that AAV9 has had that actually launched the field of neurological gene therapy, or at least gave it a huge push forward. So it's probably one of the simplest virus, barely one actually. Sometimes people jokingly call it an almost a virus. And the reason it has only 2 genes, encodes, we knew for many years they encoded 7 proteins. Now we actually know that it encodes 9. And it really is a nanoparticle, 20 to 25 nanometers, so tiny. And of course, it also comes with limitations that the packaging capacity, the amount of DNA that it can carry inside is also limiting, right? So one of the important aspects, so there are 2 aspects that are important, is that it doesn't replicate on its own. And so it depends on co-infection with other viruses. Obviously, adenovirus, I mean, you saw that's how it was discovered, but it also can replicate in the presence as HSV. Pretty much everyone in this room, or maybe 50% of us, have either been exposed to it, because we'll have, at least the adults, 50% of us will have neutralizing antibodies to one form of AAV or another. So we've been exposed, most of the, a huge number of humans. And in fact, there's really no association of AAV with any disease to the best of our knowledge so far. So that makes it for an interesting platform from where to derive a gene transfer vector. And, it took us about 30 years, and sounds like a lot, it is a lot, but how to figure out how to take out all the genetic information of the virus and replace it with what we want. And that's just, a sort of standard cassette that you see up there. Here you have a promoter, your gene of interest. Of course, we call it gene, but really it's not a gene. It's, it's a cDNA, so it's an abnormal RNA in some ways. And have a poly-A. And the virus, the only things you need is these tiny little elements that are non-coding, that are at either end. They're called inverted terminal repeats. So why do we like it? Why do we like and use so much the virus? I mean, a simple reason. It's the most efficient platform that we have for in vivo gene delivery, at least so far. We used to say that it doesn't integrate into the genome. Well, I'm not quite sure. It actually does integrate, but at low efficiency. But perhaps the most important aspect, at least in non-dividing tissues or slowly dividing tissues, is that it can mediate very long-term gene expression. Certainly is not the magic bullet for every application. It's absolutely the wrong entity to use for bone marrow modification, because as Blair said, bone marrow replicates and it gets lost. So certainly you have to think a little bit about what to use it for. And certainly what we've learned as we move towards clinical trials is that, humans actually don't like too much to get a viral load that we actually didn't evolve to get. It's as simple as that. In some dosings that we're doing, we're injecting about 160 times more viral particles than we have cells in the body. If you ever had a normal infection with that amount of viral load, you would be dead in about 5 seconds. Okay, so we didn't evolve to have such load in our bodies. So it turns out that unlike mice, and mice really lie in many ways of what we do experimentally, mice are just peachy. They don't have a problem. Well, the moment we start putting in humans, the immune system came to the fore and has continued to be one of the major limitations, and something that you have to survey patients very, very carefully to avoid. Actually, it can lead to death. Okay, so if you don't control it, actually can be very problematic. I'm going to jump quite a few of these things. So actually so I'm talking about CNS only today. I mean, there's obviously many applications that are being explored and quite a few products approved now. And this is just the flimsiest of barriers that makes it very difficult for us to be able to actually deliver genes or viruses or liposomes or whatever you have it directly into the brain. This is actually a plastic cast of a mouse brain. And this is just to tell you that we actually already have the perfect delivery route to the brain, right? Which is the blood-brain barrier, or the blood vasculature in the brain. And it's really remarkable because every cell in your brain has to fit in this space between the vessels, right? And it's absolutely remarkable that they actually fit. So what that tells you is that every cell in the brain is within about 10 to 20 microns from a blood vessel. So we do have the perfect delivery system. The problem is that the brain really likes to live behind a barrier in a very controlled microenvironment, and as we have evolved as mammals. Actually, this goes back in evolution quite a bit. A way in which a very tiny barrier really is like 2 or 3 cell layers actually that prevents any macromolecule from crossing the blood-brain barrier freely. Of course, we do have molecules that do that. For instance, to ferry iron to the brain, we have a receptor system and transporter that does that, but that's how we evolved. Anything else is excluded. Okay, so this is obviously a limitation for all of us that work in gene transfer, gene therapy for the central nervous system. Ways to get around it. I mean, I guess you've already seen that intraparenchymal injections is one possibility. However, that has limitations, right? So for diseases that are focal, that is certainly an application that was where we started in this field and continues to be valid actually. For diseases that are focal, this is one solution, right? But for diseases that have a global expression, and we know that the cortex in principle is what distinguishes us from other lower animals or lower mammals, the surface area is absolutely massive, and the geometry of it is devilish to figure out how to deliver it by direct injections, right? So other delivery modalities have been explored or are being explored, such as direct infusion into the CSF. And the claim to fame of AAV9 is actually, to a certain extent, it can cross the blood-brain barrier. And that's essentially the principle that was used to develop a therapy for SMA. And it does work pretty well. I mean, it's not perfect, but it works pretty well. So these are the different modalities, and now I'm going to talk to you a little bit about our gene therapy program. When I started this program, honestly, I knew absolutely nothing about GNAO1. I had never heard about it. Of course, I had heard about the G proteins and G protein coupled receptors, but that's really where my knowledge stopped. And so the process that we usually use in approaching any new disease that unfortunately we get contacted very often from parents with, that their kids are affected with all sorts of diseases that sometimes almost sounds like a joke, the name of the gene, because it's a soup of letters that, which really makes no sense, at least on the surface. And the first step that we do is to try to understand, where is this gene expressed? Obviously, most patients come to us with neurological diseases, so obviously expressed in the brain. But we go and look at where else does it get expressed? Where do we have to think of it not just as a CNS disease, but what else needs to be fixed, right? The other aspect is that, we like to think about genes that, it's all very simple, but actually we achieve complexity in biology by changing the ways in which genes are expressed and different isoforms by alternative splicing. So there is a lot of databases that can be used that can tell you the different isoforms that have been documented. GTEx is one that is across the river at MIT that gives you a little bit of that information. And one of the things that I was not aware at all was about the GNAO1A and B, and here is the, the protein homology between them, and they really differ only in the C-terminal domain. But one aspect about biology is that we don't need too many amino acid changes to actually change the protein function. So 20 amino acid changes, while it is very small, biology in general, we're economic. Evolution works in fairly economical ways. And if we actually express two genes, presumably has some function. Now, whether we understand it or not, well, that's an entirely different story. So what I started by doing is that I wanted to know, first of all, where are they expressed, at least to gain some knowledge, mostly for myself than anything else. And what we did was actually look at RNA-scope, that is just looking at where the RNA is expressed in an adult mouse brain, and probes that are specific for each isoform. And not surprisingly, in most cells, it's co-expressed in most cells, both isoforms. But interestingly enough, actually, there are some areas where they're mutually exclusive. And we can see one example here. Oh my goodness. On the hippocampus, where the dentate gyrus is clearly expressing the GNAO-B. I called it 1.2. I apologize to the people that actually know more about this than I do. And then you can see also here in the olfactory bulb, also another area, neuronal region, where again there is this expression. So what we don't know really was what's the function. So I'm going to go back a second here, is that in our studies we decided to test both isoforms, just in case actually one of them worked better than the other. And actually I think they do, and it actually agrees with what everybody has said in this meeting today, that actually A is probably more important than B. And then we also had designed some bicistronic vectors that actually encoded both of them. So the biorender, very useful. Actually, we all use the same pictures, but this is the two paradigms that we started with funding from the Bow Foundation. This was done in Rick Newbig's lab, where they tested bilateral striatal injections. And as you've seen before today, multiple times, the open field test is the one that used, that after all, he had characterized the R209H mouse model. And this is now published, and the results, essentially what it showed is that, the hyperactivity or increased activity in the open field, there was a little improvement, a little decrease after treatment with the AAV therapy. I took a slightly different approach. I wanted to know that, what happens if you actually deliver the gene globally, and to achieve that in the most efficient manner, and I'll have to admit, fairly artificial, in the way that if you do a neonatal intracerebral ventricular injection, meaning putting the vector into the lateral ventricles into the CSF, you can actually distribute the gene very widely throughout the brain. The reason that it's artificial is that this really doesn't occur at any other stage in development in a mouse, or for that matter in any other species. So you can get 50, 60% of the cells genetically modified. Usually this is where I start, because if we fail with this, we know that any other modality is really not going to work either. So we're starting in a very reductionist manner approach, that this is where we can exclude very quickly what we're doing, if it's useful or not. If it's useful, great. We can then explore other later delivery, later stages. Unfortunately, I'm going to go quickly through these slides, because what we found is that unlike the striatal injections, the ICV injections had absolutely no effect on the model. It did not change at all the differential in hyperactivity. You can see that here. And the only statistically significant that really doesn't matter, actually, is that the difference between wild type and heterozygotes injected with either vector or PBS remained the same, meaning that there is really no effect whatsoever. There is multiple parameters here that we can go through all of them, but the graphs are essentially replicas of each other, and just looking at different parameters that you can look in the open field, different types of parameters that you can measure. So we really didn't find anything. At one point, we're kind of hopeful that this meant anything, that there was no difference between the wild type and heterozygote mice for this vector. But unfortunately, there is no statistical significant difference between these two groups here, between the ones that are treated with GNAO1 or GNAO1A vector and PBS. Unfortunately, despite this statistically not finding a statistical significant difference there. So the question then was, well, are we even expressing the protein, or even making anything with this vector? Are these vectors completely useless and non-functional, right? I mean, it's a fair question. So, we looked at the RNA level. We looked at developed probes that are specific for mouse and specific for each one of the isoforms that we're expressing. what we show is that we don't really see difference between groups. There's a couple of mice here that we don't really have an explanation why they're down there in terms of levels of the endogenous mouse gene. And what we see is that, yes, we can actually express both genes. And here the fact that this is lower, reads lower, it really is related to the probe that we haven't fully optimized and validated the probe and its efficiency. Clearly, this one works extremely well. This one here, there is a big difference in terms of efficiency of the probe. So I don't think this is meaningful, this difference that we've shown here. These are some experiments that we still have to do. Protein level, this is something that I've heard from multiple people that, again, surprised me. This is the only time that I've ever seen this in my life of looking at many, many gene therapy programs, is that we cannot see an elevation in total amount of GNAO1 protein. And so the levels are essentially comparable between the PBS groups and the treatment groups, both in males and females. And we can postulate many reasons why this could be the case, but really this is the only case that I've ever seen where we can't actually overexpress one of these proteins. And unfortunately, we don't have antibodies. If anyone has them, please do let me know. That can distinguish between the human and the mouse. So we're kind of stuck with this antibody that recognizes all of them. So not terribly useful to ask this particular question. So unfortunately, the conclusion out of this study really is that it makes absolutely no difference, this treatment modality, this global delivery in terms of the phenotype that we have, at least for this mouse model. Certainly the vectors seem to be functional to some degree, that the bilateral striatal injections do have an effect. And we generated thought that perhaps doing a combined treatment, sequential treatment, could actually enhance the effect. That you need a striatum to have that primary effect, but also delivery elsewhere would enhance, magnify the effect of the treatment that we were doing. So we treated the animals neonatally, so when they were born, literally at the day of birth, waited for 8 weeks, looked at the open field. What did they do? Meaning, are the results still comparable to the initial study that we did? Waited for 2 weeks, did the bilateral striatic injections, and they waited for another month because that's the paradigm that had been used by Rick Newbig's lab, and did the open field test again. So again, very complicated graph, and I apologize for that. I just didn't want to make 500 slides. Use the slides really that what it shows is that there is no difference with this approach. So the story is really pretty embarrassing, actually, or at least to a certain extent embarrassing, that it led us to actually repeat a little bit of the study that Rick had done, just that, to prove to ourselves honestly that we actually know how to do striatal injections, because, it's something that we've done for many, many years and a bit surprising. So we did that study, and in fact, yeah, we actually do see a difference. So I don't quite understand what is the difference between what is the effect of delivering these GNAO1 early in development, so at P0, and then coming back and doing the striatal injection, not having an effect. But if we deliver early in development PBS, so just a vehicle, no gene, but come back and inject bilateral striatal injection, we can actually have an impact in the activity of the mice. Other parameters we don't really see, at least a statistically significant impact. So these are some of the questions that remain a bit of a puzzle to us. And part of it, one of the issues that we have, and all these things are here, of things that we're doing, is that, we really have no clue, at least I don't have a clue, what are the structures in the brain that drive these phenotypes, right? I mean, hyperactivity is not necessarily a traditional motor component. I mean, looking at the patients certainly does not look like that's necessarily an issue. Certainly seems to be reproducible across multiple mouse models, this hyperactivity. But, is the striatum, I mean, this is the first time I saw the presentation about the activation in the striatum only, suggesting that in fact the striatum really is the driver. Well, I guess this is to be done. So, we're looking for better models, honestly. We need more robust models. This is very, in my mind, that difference is relatively subtle. I think having models that are better ones, so it's great to know that people are actually working on that. And, I just want to finish by acknowledging, of course, Rick Newbig is our collaborator on the initial project, on this one as well. And this work was done by Tyler mostly. I mean, Tyler is over here attending this conference, and he's worked tirelessly to make this happen. And of course, had some help from other people. And then Rita, we've worked together for many, many years, and has been instrumental in being able to do that. And of course, we couldn't do it without the support from the Bell Foundation. So thank you for your attention, and happy to answer.
Thank you so much. Time for questions, all right.
Thank you. So for the Western blots, basically my question is, have you considered driving expression of a tagged version of G-alpha-O so that on this Western blot you would be able to distinguish the human misexpressed isoform and compare its intensities to the endogenous levels?
We do have the constructs. We didn't use them in the studies. There's always the concern of any impact in function, but we may actually have to go back a little bit and really just deliver it to a couple of animals to really demonstrate that, yes, we actually do make protein. In the paper, in Rick Newbig's paper, they actually started with that to show that it can be expressed in striatum. So we know the vector works because they've done that already with an HA tag, either HA or MIC tag. I forget which one. And yeah, they did see expression. So that's part of the reason also that we didn't repeat that aspect. But we're going to use mass spec to really demonstrate that, in fact, we can see the human protein, because otherwise, if I reviewed a paper like this, I would kind of laugh a little bit that, please show me the protein, because otherwise your vector might just not work, right? So Blair has a question back there.
I might ask sort of. Yeah, well, that's working. So one of the textbook things that you'd learn about GO, right, is that it's expressed at level of 5% of the total brain. So seeing you having hard times overexpressing on top of this, I was like, well, nature's may have done it at the maximum that it's possible. But I was wondering if you've ever compared like in a model system in HK cells, how much you express with these viruses, and load like the striatal sort of a lysate right next to it, and just to see how much you're getting.
Right. We've done the comparison. AAV9 in cell culture really is terrible. It doesn't work all that well. One thing that I can tell you that I actually forgot to put on the slides and the dosing, I apologize for not putting the dosing here. We actually did see toxicity. We got to a dose in which the animals actually had seizures, which is, so clearly expression levels can get to a point that is problematic, right? Obviously, we dosed down to do these experiments. That was for the striatal injection. So we actually did see animals seizing from the high dose that we injected. But your point is, thank you for the suggestion. Appreciate it.
So first, two questions. The first one's just a technical one. There's different AAV9s, and was this AAV9 PHPP? There's many different.
Yep, this is the good old one.
Okay.
Yes. Yep.
Yeah, we had seen some really beautiful delivery after intraventricular delivery with PHPV, and I don't quite understand why, but both in adult mice even and in non-human primates. Do you think it's a distribution problem, or do you think it's?
I think it's is a targeting issue with the ICV delivery and a combination of the capsid and promoter. So we like to say and state that the CAG promoter is a universal promoter. Yes, to a point. Yes, it does express in many different tissues, but if you go through the literature and look carefully about gene transfer to the CNS, it really is not a universal promoter. I mean, people have said for years that we could not transduce oligodendrocytes. Years. People changed the promoter from a CAG to a myelin-specific promoter, and all of a sudden, oh my God, AAV9 actually does transduce oligodendrocytes. So in the field, we tend to conflate the two things, and we can only know what we can see, right? Understandable. But so there is that aspect there that I think can confound the results here a little bit as well.
I guess just the follow-up would be if it wasn't a problem with delivery and distribution, and it was, you're getting great levels in both the early, postnatal, and late, could this just be that this mutation is having a differential effect in development? There's still brain development, at P0 in mouse, versus in the adult brain. So if it's, you know, a loss of function later in life, you're correcting for that in the striatum. But if the developmental effect of this mutation is a gain of function, now you're worsening it or not improving it. And we know that each of these mutations have very complex pathogenic,
Great
probably
point.
over-interpretation.
No,
but
no,
it's
not
cool.
at all. I mean, it's a great interpretation, actually. Thank you. Yeah.
I think it's time to wrap up. Let's thank
Thank
Miguel
you.
and all of the speakers of the session for this fantastic series of talks.